I'm Chris Rourk. I'd like to first thank the conference organizers for allowing me to speak, and all the other presenters. I've learned an enormous amount listening to your presentations. Thank you. I'm going to be talking about the catecholaminergic neuron electron transport action selection mechanism hypothesis. I'm reading as CNET for time saving. So I'm going to talk about the hypothesis that was pretty much presented for the first time in 2018, some of the predictions and the evidence that has been accumulated since then, and then some thoughts on how to include this in models of consciousness. The certain CNET neurons I'm going to be referring to, or at least encompassing, are the substantia nigra pars compacta, or SNC, which generates dopamine, and it's provided to the striatum, and the locus ceruleus, or LC, which provides noradrenaline to the frontal cortex. There may be other catecholaminergic neuron groups that are affected by this mechanism and be talking about, but these two definitely appear to have neuromelanin and ferritin concentrations that could be implicated. And I would just note the frontal cortex connects directly to the striatum, and it also connects to the LC. The SNC is viewed as being external to it. The SNC and LC are viewed as modulatory signal makers. They don't provide excitatory and inhibitory signals, but modulatory signals, at least in this diagram. So CNET was based on this observation about ferritin and neuromelanin, and why? Well, because ferritin has been studied in solid state implementations, and they've observed electron tunneling in association with it. And in electron tunneling, the electron becomes coherent, and then it localizes in a different physical location. So it's tunneling or moving location. This is one of those mysteries of quantum mechanics that are very interesting. So this has been observed by many researchers. Neuromelanin, no such observations, but it has other interesting physical properties that I won't get into. And what I didn't know is that there was enough ferritin and neuromelanin in these structures to transfer energy, because these structures these quantum dot is what they call them, structures can transfer energy. It would have to provide a switching or routing function, because otherwise the energy just goes anywhere, and it would have to be presumably transferred to a specific neuron or group of neurons to help them reach action potential for action selection. So why action selection? Well, the large SNC dopamine neurons have extensile axonal arteries. Most of the SNC neurons, about 99% of them, are small, they're tonic, but the large ones are phasic, which is more associated with non-automatic activity. They're also, the SNC is at the top of the D1 or direct pathway, which is associated with action selection. An interesting thing about these neurons is that they connect to cortical axons at striatal dendrites, and this causes those SNC neurons to fire. And that's unusual, because usually neurons fire from dendritic stimulation, not from axonic stimulation. They're extensive enough, with over upwards of 1 million or more synapses, to coordinate action selection by stimulating the striatum. An action selection mechanism would help prevent seizures if they are associated with action selection, because otherwise, if they could fire simultaneously, you would have multiple different actions trying to be selected, and a seizure could result. An interesting thing to note is this could integrate cortical signals when no action is being selected. So you've got 5 to 10,000 of these large neurons with a huge number, maybe upwards of several 100,000 cortical signals coming in. And all this is happening while nothing is being done, no action is being selected. So this could explain how these signals are being integrated. When you talk about integration, I think you have to be a little bit more specific by what you mean, because integration means many different things. Now, however, I was kind of going up against the conventional understanding of these large SNC dopamine neurons. In predictive reward signaling studies, the understanding was that the phasic dopamine response does not code movement. And they made that because it seems like it would code movement, but according to the predictive reward signaling, it doesn't. Now, predictive reward signaling is you make an action, and then you see a reward, and if it's better than expected, you get a dopamine signal. And that's what they observed. But the way they did that is they put microelectrodes into the SNC, and they made measurements, and they generally observed that. For these two examples here on the right, you can see that there's a significant dopamine response prior to action selection. Now, is that because the monkey that they're testing saw after the door opened a greater than expected reward? or is it because the monkey decided to move his arm into the box? It's not really terribly clear, but there's at least some evidence that there are two different functions going on. One is action selection, and one is predictive reward signaling. And the CNET hypothesis, in conclusion, says that these neurons are all connected through this electron tunneling mechanism, and that this tunneling mechanism directs the current, the electrons to the neuron that's best situated to fire. This is consistent, though, with the predictive reward signaling studies that show that dopamine neurons are activated by rewarding events that are better than predicted and depressed by events that are worse than predicted. So as you can see in the striatum, the cortical signals come in, the cortical processing says, hey, this is a better reward, better outcome than we expected. More signals go to certain neurons than others, and again, that's just a neuron function as opposed to predictive reward signaling versus action selection. So a number of predictions were made by this hypothesis where there wasn't really very good evidence. I'll just go over those really quickly. Electron tunneling in substantia nigra tissue. I'm probably the first person to ever have this done. It was conducted at EAG labs down the road in Sunnyvale. In the bottom right-hand corner, you can see the expected result, which is only negative currents based on the convention being used. This was in a control sample with no neuromelanin or ferritin. In the top right, you can see a substantial widespread positive currents, which shouldn't happen under this testing. I mean, it just shouldn't be possible unless there's electron tunneling. That's an 80 micron square sample. If you increase the resolution down to 2 microns square on the left, you start to see features coming out like neuromelanin organelles on the right of that picture and layers of ferritin on the left. So this was good image, good research. $4,300 is what it costs for me to self-fund this test. Not terribly expensive. If somebody wanted to do research, they could further investigate the electrical properties of these tissues. This was the evidence I had in 2018, the best evidence that showed kind of arrays of ferritin between neuromelanin organelles. This is an electron microscope. They used little black dots of immunogold markers that were tagged to ferritin. So this shows that there's at least some ferritin, but these black dots could be other stuff. And it's difficult to tell. And on the left, I have an image of ferritin. It shows it has a protein shell, and inside is a solid state material, ferrohydrite, which is different from most biological materials. The electron spectroscopy image on the left shows layers of iron outside of the neuromelanin. So most iron in this SNC is ferritin. So this kind of provides some better evidence that there are layered structures outside of the neuromelanin organelles consistent with the electron microscope image. On the right, there's X-ray imaging technique that shows iron in the glial cell areas between the large dopamine neurons. This doesn't quite get us to arrays though, so I kept looking, and I found in placental cell tests, there was increased magnetic ordering detected under small angle neutron scattering. So on the left, you can see this electron microscope image shows pretty good order in these placental cells. In the middle is the small angle neutron scattering magnetic ordering. In the tissue, it's really high. Outside of the tissue, when they extract the ferritin and just test it in bulk, there's almost no magnetic ordering. So it's kind of weird that this ferritin in the tissue is behaving like a quantum dot system. Now, I'm not going to talk about quantum dots, I'm sorry, it's just too much physics to get into in a short period of time. On the right is an image I found of a glial cell in an SNC, which shows these little black dots that look suspiciously like ferritin. The paper doesn't say that they're ferritin, but I think that there's at least some data that shows that there's possibly these arrays of ferritin between neurons. And this is another study that shows ferritin being studied with small angle neutron scattering and behaving like a quantum dot solid. So electron tunneling through these arrays that look like they exist would have to occur in order to get the electrons from one soma to another soma, because they're separated by microns, and that's just too far for one tunneling event. Tunneling is nanometers. Professor Competia published some results in 2019 that showed in a layer-by-layer device that there was tunneling over 40 microns at 3 volts. Ferritin is a nanoparticle, it's hard to manipulate. She did this bulk layer-by-layer deposition, which provided sufficient order, at least in one of her samples. She didn't give us a lot of data of this transport. I was inspired to do some additional testing on myself, and I duplicated her electron transport results. I used a 4-terminal device. I'll talk about it in a minute. And then on the far right, Professor Middleton is an expert, one of the leading developers or innovators in understanding how electrons move through quantum dots. And he showed that there's a log linear behavior for this electron tunneling behavior, and that's consistent with both Ruka's and my results. So this is a diagram of the device that I designed. Professor Kai Shen at Ningbo Institute of Materials Technology and Engineering did the tests. We had about 32 dyes, 75% of them conducted no current. So even though you have multiple ferritin layers, ferritin by itself is not enough to transfer any energy, any electrons. It's not classical conduction. You have to have sufficient order to have the electrons tunnel through, and that's what we measured in the other 25%. And in some, we had some very clear indications, like the one on the right, of a routing or switching mechanism. And what's basically happening, I'm going to try and do this without changing the slide, is that the electrons are flowing from here to here, but they're not flowing from here to here. And that's weird. That's a solid state, unusual behavior for electrons. I determined it was probably Coulomb blockade behavior. I'm not going to get into that. But I do note at the very bottom there, this excellent agreement between the experimental data and the Coulomb blockade theories demonstrated. That was Professor van der Zandt, who did tests at 4 degrees Kelvin with single electrons, where he could say pretty clearly that there's Coulomb blockade behavior. My tests are at room temperature, under ambient conditions. You can't do single electron tests under those conditions because there's just too much noise, so you have to look for bulk transfer. So getting back to the fifth prediction, this action selection, and the modulatory understanding of reward dopamine signaling. In 2021, Professor Pascal Kaiser at Harvard published this great paper where he determined, after extensive work on the way that these dopamine neurons function, is that for movement control, dopamine modulates moment-by-moment activity in the striatum to mediate action selection. So again, this is a different understanding. Any neural model that's based on a modulatory-only behavior of these dopamine neurons may be wrong if this is right. It's based on a reward signaling model and not on an action selection model. And earlier, if I'd been paying attention, I would have found this great work. He said that Professor Kaiser said that the architecture supports spatially and temporally precise coding for dopamine. So again, the earlier modulatory understanding was that it's just broadcast, that it is not spatially or temporally precise. The reward signaling work showed that glutamate released from corticostriatal terminals controls dopamine release. That's weird. It shouldn't control dopamine release. It's being provided to synapses at the ends of the axons, nowhere near the soma. So this is unusual behavior. Actually, Professor Schultz looked for various ways for these signals to get conducted back, and I wasn't able to find that any of them were ever shown. Similar varicosities are seen in the locus coeruleus, and spatiotemporal activity in the targets in the locus coeruleus has been shown. These are bilateral structures, the LC and the SNC, and when you damage one side, you get this either behavioral anomalies or working memory loss that resolves over time, which suggests that it's programmable. You know, I kind of skipped over this, I'm sorry. Only about 20% of these varicosities are active. So they're all next to cortical terminals, but they don't become active until there's some cortical programming that goes on that says, hey, this movement would be, it's probably muscle memory, might be what it is, but in any event, it shows that this is very similar to a programmable structure. So how do we include this very complex neural structure in a model of consciousness? And is it even consciousness? I don't know. More work has to be done, but it could be. It seems to be related with this experience of seeing something and, oh, a lion comes in the room, I better run out that door. Or, you know, everything's fine, I don't need to move. And you could use a neural network to train, you know, this is a big switch. It's got an unusual function. So prior to understanding how the switch itself works, you could train a neural network if you knew the state of all these synapses, and you could use that to predict individual action selection. That's probably a lot of work to map those all out. Neural networks have input layers and output layers, so you'd be looking at an input layer with, you know, millions of inputs and an output layer of several thousand. And you have to just model all this. You have to have the actual data to train the neural network with. It might be possible to get like familiar inputs, primitives of like, for example, if you're driving down the road, speed, obstacle in the road would be the inputs, outputs would be pressing the brake, turning left or right, doing nothing. And you could possibly train an action selection neural network in this manner to create a conscious selection. Now, there have been some references to, well, I don't remember driving down the road, so I wasn't conscious. But what's really happening is, I think, that you don't remember it because there was nothing happening, there was no action required, and there was no memory being generated. It doesn't mean that you weren't conscious, it just means that you don't have a recollection of that because it was uneventful. CNET could be included, I think, rather easily in the global neuronal workspace. They currently use networks, neural networks, real neural networks, not artificial neural networks, but it is a modulatory signal. So to the extent that a GNW model has a modulatory signal, it's not accurately representing or simulating action selection according to this recent work by Professor Kaiser. In this recent paper, you see the dopamine neurons down at the bottom, the cognitive level. I think they would be more in the conscious level. The cognitive processing precedes it. I mean, Adam Saffron was mentioning his tests that show that there's some cognitive processing that is measured that's supposed to destroy free will. I disagree with that like he does, but I won't get into that right now. On the right, we have a model that used behavior modules, action selection behavior modules on the left as inputs to a workspace module, and it used this GNW type process for controlling a computer game character. So, again, I think that works, but this kind of recognizes that you need an action selection module, but there's no action selection inherent in GNW as it's currently formulated. Probably the more accurate place to put it would be inside of the workspace modules and not off to the side. I'll try and just wrap it up here. And special thanks to everybody who's helped me as a citizen scientist, self-funding all my research. I do appreciate any help I can get. I know it's a lot of work to try to understand this very unusual idea.